All right, Philip Ball, thank you so much for coming on the podcast.
Oh, it's a pleasure. Thanks for inviting me.
So, we're here to talk about your book, How Life Works. But first, can you just talk a little bit about your professional background and some of the experiences you had in that role that inspired you to write this book?
Right. Okay. I've wandered into being a freelance science writer. It feels like a very sort of random trajectory, but in some ways it feels very natural that I've ended up here. So I began my scientific training taking a degree in chemistry. Then I followed that up with a PhD in physics. And from there, I went to become an editor at the science journal Nature. And that was really my sort of point of entry into science communication more generally. So although as an editor, I didn't have explicit sort of duties in terms of writing, there were always opportunities to do that. And I was interested in writing about science. And I just did more and more of that until I realized that I couldn't fully fulfill what I wanted to do in science writing and remain an editor of Nature. So I gradually stepped down there. This was about 20 years ago when I finally became fully freelance. And that's what I've been doing ever since. So I write about all areas of science. Some tend to capture my interest and I sort of stick there for a while. And that might be quantum mechanics, that might be topics in the history of science, or with this book, it's been trying to find new and I hope better narratives to talk about biology, and in particular to talk about the question of how life really works, how our cells from the molecules upwards, how they really do that job. And I guess that's really a question of trying to find new ways of talking about what life is. So the book is really an attempt to do that. It seemed to me that the stories we tended to tell about that were often they go back to the 1990s, 1970s, 1950s even. And, you know, the science has moved on. So I think it's time to find new stories to tell about life.
Right. And one of the themes you hit on in the book is that we've fallen victim a little bit to a psychological tendency, I guess, to oversimplify things. And our understanding of biology is sort of kind of a reductive version of what actually happens. Is that right?
It is. And in a sense, that's always the case in science. You know, well, very, very often we have to find simple narratives that give us some sort of foothold to our understanding. Whether, you know, that's whether we're teaching it to school children or to undergraduates or actually doing the research itself. You know, there are kind of simple stories that we tend to tell. And I think the one in particular that has arisen in biology really over the past half a century at least or more is one that takes the view, I suppose, that life is basically encoded in our genome. It's encoded in our DNA. Somehow within there is all the information, the program, if you like, that runs the cell and runs the organism. And in a sense, all that then needs to be done is to read out that program. And that's what life is about. The more we've come to understand about not just what genes are, but what the whole of our genome, all of the DNA that we have, which is more than just the regions we call genes, what that does and the molecules that it produces and how they interact and how then cells interact and how tissues are built, the more we've understood about that, the less clear it is that that kind of narrative, that sort of bottom-up narrative that it's all coming up from the genome and it's just reading that information out, that seems less and less satisfactory. So I wanted to try to explain why that was so, what is valuable in the old model, because, you know, always in science, the old ways of looking at things, you know, they usually have something valuable that's worth retaining, but what we need to change also, what we need to add to that narrative to understand this process of life.
To trace this a bit historically, how does that DNA-centric view of biology, that our DNA is the sort of blueprint of biology, how does that perspective intertwine with some of the sort of societal changes that maybe happened over the last 100, 150 years or so?
Well, I guess typically the DNA revolution is traced back to the discovery by Francis Crick and Jim Watson in 1953, which of course was building on other people's work, Rosalind Franklin and others, the discovery of how information can be encoded in DNA. And it was really that discovery that verified for the first time. Until that point, people weren't even sure that our DNA was where our genes reside. For a long time, people thought genes might be some kind of protein molecule. So that was really the beginning of the modern era of molecular biology. And What's striking about it is that it happened exactly at the same time that digital computers were starting to become a reality. And very often the information in those computers, I mean, I'm old enough to remember this from the 1970s at least, that the information was encoded on magnetic tape. And it seemed an extraordinary parallel that, that's where the biological information seemed to be encoded in this tape of DNA. It was like the binary code that is used for, storing information in computers, except that it had 4 letters instead of just these ones and zeros, these 2. But the information was stored there in what seemed like linear fashion, and it was being read out in much the same way that computers read out that information and run a program. And, all of that, it just made for such a neat story that, it was irresistible, really. And so that was really the picture that dominated, really up until the Human Genome Project, which began in the 1990s and started to sort of wind up in the early 2000s. And, the idea was that, okay, all we need to do is to figure out what that information means. And that was really what the Genome Project was about. It was about reading every one of those. There are about 3 billion of those letters, if you like, in the entirety of human DNA in our genome. And the hope was that once we had read it, then gradually we'd sort of figure out what it was actually telling us, what it actually meant. So that, I think, it seemed like such a neat story, and with such a clear parallel to the emergence of information science quite generally, and of information technologies and of computation, that, it was an irresistible story to seize onto. And it's not that it's wrong, but that's not all of the story. And in fact, the more we understand about what is actually encoded, because it really is encoded in our DNA, the less it has seemed to fit with that picture of just a simple readout of information that goes to sort of, build cells and build organisms. And the way I try to rephrase this in the book is really about there being, for complex organisms like us, which are made of, billions and billions of cells, all with the same DNA, but very different types of cells, that there's really a hierarchy of processes and of levels at which things are happening, each of which has its own rules that depend to some extent on what has happened on the levels below, but is not dependent on the fine details of those. There's a sort of autonomy in each of those levels, whether we're looking at the networks of interactions between DNA and proteins and the rest, whether we're looking at the way cells interact, whether we look at the way tissues are built. At each of those levels, there are rules that are unique to that level. And that's really what we need to understand how a complex organism like us works. We need to understand each of those levels and not to give any of them a sort of privileged status over any of the others.
Right. And one of the examples you use in the book is that we don't have a gene that says we're going to have 5 fingers. There's not like a 5-finger gene. It's just that through a chemical and thermal and sort of electrical processes, fetuses, when they grow in the womb, because of all these different networks, as you say, they tend to grow 5 fingers every time on each hand. And one of the examples of that you talk about are gene knockout experiments. Can you talk a little bit about the complexity that some of those experiments have revealed over the years?
Okay, well, the simplest picture that we had, way back, maybe in the 1950s, 1960s, of how genes would work, tended towards the idea that there would be particular genes that were responsible in some way for particular features of the organism. Now, I mean, even then, I think, before we even knew the genes were DNA, there were signs that actually it wasn't going to be quite as simple as that. But to an approximate degree, that was the kind of idea. And you talked about the notion, although it's an untrue notion, that there might be a gene for 5 fingers. So people talk and still do talk actually about a gene for this and a gene for that, a gene for ears, or a gene for teeth. And in general, there is no such thing. There is no meaningful way of talking about genes in that sense. All genes, or in fact, the conventional idea of genes, and even this has been complicated in recent years, but the conventional idea is that what a gene is for making a protein molecule. And these proteins are often, in general, they're enzymes, they're catalysts that allow the cell's chemistry to happen. So we can say there is a gene for making the catalyst that, digests lactose or something. But we can't say any more than that about the role it plays in our shape and in the development of our body. And in fact, lots of genes seem to have lots of different roles in lots of different processes. And, that's the thing that has really come out in the past 20 or 30 years. And if we look at just about any trait we have, whether it's behavioral or physical, we can find genes that are correlated with that, so that if there's a change in the genes between different people, there's going to be a change in those traits. But very often those traits, if we think of something like height, there might be hundreds, even thousands of genes that seem to have some involvement in that. Now, we only have 20,000 protein-making genes in our body. So, if 1,000 of those are involved in height, for example, that's a big fraction of our whole genome. So we can't do that easy mapping. And what that often means is that researchers might think, well, here's a gene that does something really crucial to the body, makes this enzyme that seems to be really, really important for how the body works. If we use biotechnological methods to knock that gene out, to basically silence it, stop it making its protein, and this was something that became ever more possible since the 1970s, then the organisms should just die. They should just be not viable. And occasionally that's true. Occasionally the result of knocking out a gene like that is one that you don't expect at all. Very often the result is nothing that you can see. The organism just goes on living as happily as before. And you think, there must be something different, but we can't see anything. So that was really what started to give us some intimation that this simple picture between genes and what we call the phenotype, the traits of an organism, that it's really not as simple as that. And that's really what started to worry me when I started to see papers like this appearing and being published in Nature when I was an editor there in the 1990s. Often the biological editors would come along and say, we're publishing this paper, about this gene X that we thought was involved in, I don't know, it could be cancer, or it could be the development of, the nose or something. And hey, it's involved in something totally different as well. And, eventually I plucked up the courage to say, what does this mean? And the answer I got was, we don't know. And that made me start to think, well, are we telling the right story? Are we telling the public the right story about genes when we give the impression that there are genes for this and genes for that? And it's very clear that now, we need another way to think about the relationship between genes and the organism as a whole.
Further complicating the importance of DNA is epigenetics, which when I first started learning about sort of blew my mind and definitely does not necessarily fit into the narrative that DNA is the sort of all-important blueprint. You can correct me if I'm sort of paraphrasing this incorrectly, but in the book you say that the real sort of agency of genes are less sort of what is transcribed and more about when and where they're transcribed, which is partially determined by epigenetic factors. Can you talk a little bit about how epigenetics works, essentially, or some of the different sort of epigenetic concepts? And then to what degree there's evidence for the fact that the possibility that epigenetics might be sort of inheritable across generations, definitely in plants, but also potentially in animals as well.
Yeah, okay. So epigenetics, it's actually an old idea. It became very trendy about sort of 20 years ago, and you were hearing about epigenetics all the time. But actually, we've kind of always known, since we've known about genomes, that there must be something like epigenetics going on. And that word was certainly used, at least in the 1940s. I mean, actually, epigenesis, which is kind of what it's derived from, goes all the way back to Aristotle. But the idea is that, as I say, in an organism like us, we have lots of different cell types. Some cells become liver cells, some become heart muscle cells, some become skin cells. Every one of those cells has the same genome. And we've known that for a long time. So there must be something other than the genome that is determining things like the type of cell and the type of tissue that gets made. So that's really what epigenetics is getting at. And, that obvious truth in our development means, as I say, we must have known something like this was going on for a long time. It's only recently, though, in the past several decades, we've started to understand what it is that causes that. And it's basically about what really matters for organisms like us and for our development from a single cell. What really matters is not so much what genes we have, but how they're used, how they're turned on and off, which genes are active, that is, are being translated into their proteins or other molecules in particular cells at particular times, and when that is happening and to what degree that's happening. That's really the crux of the initial process of, going from genes to an actual cell that does something and does something specific. So we started to understand how that happens. And it happens in a variety of ways, all of which have kind of come under this term, this umbrella term, epigenetics. So, for example, for some genes, there are enzymes that will literally stick a bit of a molecule. It's actually like a little bit of a methane molecule, stick it onto the DNA itself. And they might stick, many of them on the DNA get sort of dotted with these little so-called methyl groups, and they will influence whether the gene is expressed or not, which means whether the protein or whatever other molecule it encodes, it might just encode an RNA molecule, whether it gets made. So that's one way. Another way is that the DNA itself is wound around little kind of cylinders of protein called histones. And there are some enzymes that will stick molecules onto histones, and that will also influence it. But then there are ways that the DNA itself is packaged up into our chromosomes. And those are within a cell, they're kind of packaged and folded and so on. And there are enzymes that will affect that will unfold bits of the chromosome or that will pack it up again. And whether or not it's packed up or unfolded will determine whether the enzymes that actually transcribe the gene and make the protein, whether they can get to it. So there are all these ways in which genes can be turned on and off. And, that's what we've started to understand. But it's a really complicated process. It's very contextual. It makes use of all the sort of information that the cell is receiving to decide which genes to turn on and off. And Andrew, you alluded to the idea that has been talked about now of whether some of that labeling, some of that turning on and off of genes might be inherited. So that something that happens as a result of the environment, because, literally environmental things like diet can affect whether genes are turned on and off, whether that information can actually be transferred between generations, which looks something like the idea of inheritance that Jean-Baptiste Lamarck suggested in the late 18th century, early 19th century. That is, it's a non-Darwinian mode of inheritance. And there have been some studies in some animals that have suggested that there might be some possibility of some of these epigenetic marks on the DNA getting through the process that normally strips them away when the reproductive cells are made. And so it's inherited. But I have to say that is controversial research for anything like a mouse, let alone humans. It seems there's good evidence that it happens in plants. That it is controversial still whether it happens in mammals. And even if it does, it's unlikely to be of significance in the long term, in the evolutionary sense, because it's not going to last for more than a few generations at best. But nevertheless, it's an intriguing possibility.
There's so much I want to get to, Philip. So I'm trying to move quickly through my questions. Let's talk about proteins. So there's a section in the chapter on proteins in the book called The End of Order. What is behind the sort of small but maybe growing recognition of how widespread and significant disorder is in the shapes of proteins?
Right. Well, just a scene for that. What I try to do in the book is to sort of go up through this hierarchy, starting at genes, and then we get to RNA, and we get to proteins and so on, and to try to point out at each level why the way we think about it is somewhat different and sometimes significantly different from the way we used to think about it 10 or 20 years ago. And so for proteins, one of the key ways in which that idea has changed is this notion of disorder. So the old idea that I used to be very happy with, the proteins, that, certainly was the idea up to the 1990s, was that these proteins, so they're enzymes, so they're made as sort of long strings. They're basically biological polymers. And the strings then kind of crumple up and fold up to form a very particular shape. And that determines what the enzyme does. There are little sort of well-formed cavities in the enzyme that the molecule that they're going to transform will fit into like a lock and key. And, that's the way they work. And that's how, even though our cells are full of, thousands of different proteins, they don't get sort of all mulled up. And they don't, all sort of interact with one another because they've got these very specific shapes. So they ignore everything else except the thing they're meant to interact with. So that was the nice, tidy story that we used to have. Disordered proteins have disrupted that really significantly. So we now know that many proteins, maybe the majority of proteins in humans, to a greater or lesser degree, they have disordered regions within them that are kind of loose and floppy. So they're not these tightly, neatly folded, very sort of specifically shaped regions. They're like sort of rubber bands that are sort of floating around. And this can make the proteins very unspecific in what they interact with. And it makes them, disordered regions can act like sort of vaguely sticky patches that can make, several proteins sort of cluster together quite weakly. Sometimes those patches might become ordered when they bind to another molecule. Sometimes they don't. Sometimes they remain, rather unspecific. And so the language with which proteins interact with each other and with other molecules is much fuzzier than we thought it was. And the question then becomes, well, how on earth is any sort of order maintained if lots of our proteins are just these vaguely sticky things that could, stick to each other? And it seems clear now, I think, that this is clearly an intentional thing, if you like, if you can speak in those terms that evolution has chosen, because there's more disorder in our proteins than there is in simpler organisms like bacteria. So, nature is perfectly able to make lots of well-ordered proteins, and there must be a reason why there's more of it in our proteins. And one of those reasons, I think, is that these fuzzy rules of interaction are essential for the kind of adaptability and the kind of innovation that organisms like us have to be able to do, even at the cellular level. We're used to the idea that we innovate at the mental level, that we encounter things all the time that we've never encountered before, and we have to figure out what to do. But in a way, that sort of process seems also to be happening in our cells, so that they can be responsive in various ways. You can find new pathways of interaction emerging within the cell to respond to what information it's getting from outside. So this fuzziness of the network of protein interactions seems to be an intentional feature in complex organisms like us.
Right. And so there's sort of a communications reason for having disordered proteins, because molecules at either end can be influenced by the changing shape of the protein. But if the protein didn't change shape, there would be less for those molecules to sort of react to. You talk about AlphaFold in the book, and I think when you wrote it, I believe the only model that had come out was AlphaFold 1. Have you seen AlphaFold 3 that recently came out?
Yeah, so this, I think, is the version that doesn't just predict how a protein will, well, not how it will fold literally, but the shape it will have. It also predicts the way proteins will interact with a variety of different molecules, with DNA, with RNA, with maybe lipid molecules, and so on. And it's it is fantastically impressive, the fact that we can do this. I have no doubt that it's going to be really important in many areas of research, not least of which, and the thing that excites me most, is protein design. That we can actually use it to say, I want a protein that looks like this. Tell me what sequence I need to make a protein like that. And AlphaFold seems to be able to do that. So that's really, really useful. I'm skeptical about whether, certainly at this stage, whether it's going to revolutionize drug discovery, which was the kind of message that was given when it first started coming out, because the idea was that, okay, now we can figure out what protein we need to bind to this kind of drug target. It seems the accuracy with which it can make those predictions, although it's often impressive, it may not be enough of the kind of accuracy that drug designers really need to have. It may not be reliable enough, but also that's only one aspect of making a drug molecule. There are so many more stages that you have to go through to make a drug that actually works. So, it's only addressing one part of the process. But, with that proviso, I think that it's a fantastic piece of work and it's only going to get better and better. This question, though, of protein disorder is going to be a challenge for it because, you can't predict the structure of a protein that doesn't have a structure. And figuring out what the rules, if there are rules, what the rules of interaction are between disordered proteins is going to be quite another challenge. So we'll have to see whether it helps us with the sorts of problems that involve disordered proteins.
Going back to the reductive view of biology that we tend to have, talk about how we look at the human body incorrectly as sort of like a radio or a computer, where one part sort of leads to another part, and there are all these like logic gates, and if this, then that. Can you elaborate on the sort of messiness of our cellular processes?
The picture that we were often given, certainly from maybe the sort of early 2000s, when the discipline of systems biology started to take off, you'd see these networks of protein interaction. So this protein interacts with this protein, and then that interacts with this one. And they were it was like a circuit diagram, really. And among other things, the fact that you have this fuzziness of interaction due to protein disorder and other things means that you don't necessarily have those nice, neat circuit diagrams to understand. It's a messier process than that. And it now seems that, for example, one thing that cells use is, rather than having these very specific sort of chains of interaction where this molecule comes in, interacts with this one, and then that goes off and talks to another one and so on, you get a kind of conglomeration, a loose kind of association, literally a blob, like a liquid, tiny little blob of liquid within the cell that's often called a condensate, that brings together maybe hundreds of different molecules in a vague sort of way. That's one of the examples I talk about as a kind of committee. So it's not like they form some very complex but very precise structure by all sort of sticking together in exact ways. They're just kind of all swimming around in this dense cluster that is distinct from what's going on around it. So there are some proteins that are welcomed into it and some that aren't. But somehow, out of all of those kind of, fairly fuzzy, indistinct interactions comes a decision. For example, these condensates seem to be involved in the process of gene regulation, of deciding whether to turn a gene on and off. There are bits of DNA that are involved in that process. There are RNA molecules that aren't encoding proteins. They're actually doing something in their own right. They're a crucial part of condensates. There are lots of proteins, especially these things called transcription factors, which are known to be involved in turning genes on and off, many of which will sort of bind all over the genome. There's not just one place that they'll slot into. So it's very loose and fuzzy. And, you get this kind of condensate committee formed around a gene that somehow makes a decision based on all the contextual information, really, that it has, whether to turn that gene on or off, or whether to increase or decrease the rate at which it's transcribed. So that doesn't really seem to fit neatly with that old picture of a nice, clear, circuit of molecular interactions. And we need another way to think about and to conceptualize and to theorize the way that these molecular interactions are giving rise to the sorts of group decisions that are determining, for example, whether a cell is going to become, a liver cell or a skin cell.
So with all that maybe randomness going on, how do cells sort of figure out what types of cells that they're going to become?
Right. I mean, I suppose you could say there are two parts to this problem in general for making a complex organism like us. One is the cells have to decide what type of cell to become. So they start off as stem cells that could become any tissue type, and then they gradually specialize to become more and more specialized until they reach a mature, stage like a skin cell. And the other is they have to figure out spatially where to go and what structures to make. And those two are sort of related, and they're feeding back all the time, so that, a cell that might be sort of heading one way, it finds itself surrounded by cells of another type, and it sort of changes its mind. It becomes, something to align with them. So that's a very, very complex process. But there seem to be three key types of signal that tell a cell, if you like, what it should become. So one of them is chemical signals. Things like hormones come out of one cell and stick to proteins on the surface of another and initiate some signal that is sort of sent down to the genes to decide which genes to turn on and off. Another is mechanical, that actually just pulling on the membrane of a cell can transform it from one cell type into another, or can determine what a stem cell will become, whether it would go one way or another. So, that's kind of extraordinary, and it shows that there's this mechanical language to, our development that isn't obviously genetically encoded at all, although the components are genetically encoded. And then, as you say, there's also electrical communication between cells, not just between neurons. We're used to the idea that, our neurons fire sort of electrical signals, and that gives rise to our thoughts and so on, and our nerve signals. But actually, pretty much all cells have the potential to sense one another and to communicate, if you like, with one another electrically. So there are these different channels, all of which, cells are responding to. So they're responding to their neighbors to make the decision of what type of cell they should become. And that's going on in parallel with changes to the way, for example, cells stick to one another. So there are switches cell type from being ones that are sort of well stuck together to ones that are less adhesive and more fluid, and then they can start to move. And in fact, that's what happens when tumors metastasize, because they become more fluid and then they're able to go off through the body. And that's, of course, what you want to try to avoid in that case. But during development, that kind of change to cells that are more mobile and can start to move around, that's a crucial part of development, so that cells can kind of arrange themselves where they need to be. And all of that process is clearly, there are genes involved. There are genes being switched on and off, but sometimes the structures that come out aren't really determined by, they're not sort of programmed into the genome. They're often determined by the factors that are more to do with physics. For example, cells of the same type tend to sort of stick together and form something that looks like a kind of a liquid droplet with a surface tension, and then the surface tensions determine what sort of shape it makes. So there's, that's one level of the hierarchy that is responding to signals from below and, genetic signals, but isn't solely being determined by them. There's an intrinsic set of rules that is governing the kind of structures, the kind of morphologies, the sort of shapes that the cells are forming. And it's often that is going to determine, the shape and the size that a particular tissue or an organ has.
Again, with all of that kind of randomness going on, and yes, there are these sort of governing rules, but it seems like there's just a lot of randomness that happens and then leads to certain outcomes. Let's talk about agency. So we can observe that bacteria have agency, and most bacteria are single-celled organisms. So if single cells can have agency, where does agency sort of start and stop at the molecular level?
This notion of even talking about agency in biology is still controversial. And there are some biologists, quite a few biologists actually, who don't like the idea, don't like the notion of agency at all. They feel, what is it adding to our understanding, our sort of bottom-up understanding of living things? But it seems to me and to some others that a particular type of notion of agency is central to what a living thing even is, because the question behind all of this, to my mind, is if you have this hierarchy of processes that ends up with something like us or some other organism, what is it that is keeping all of that on track? What is it that is ensuring that all the different levels of that hierarchy are doing what they need to do in ways that aren't simply being programmed from the bottom up? They're all sort of doing their job, in the right way, kind of to some degree, independently, autonomously. So, what's the sort of overall vision, if you like, that's making an organism and that's, determining what an organism then does in its environment? This notion of agency seems to me to be a useful one to be thinking about that kind of question. And what I understand by agency to mean is the ability of living organisms to change themselves and their environment, or to respond to themselves and their environment in ways that are beneficial to it, which is to say that allow it to stay alive, and then ultimately for an organism also to propagate itself, to reproduce and to feed and so on. So it's having to make choices about that. It's having to, it receives information from its environment, and it has to, the agent, the organism, has to think, is that something I can just ignore? Is that something I need to flee from? Is that something I should go towards because it's a good thing? It has all those choices. So that's this notion of agency. And I would argue that it begins with the cell. So anything, because the cell is the smallest thing that we, I think, generally agree is alive. Below the level of a cell, it's not clear that there is a meaningful definition of life. People talk about where the viruses are, and that's an interesting discussion to have. It seems to me that actually viruses become agents when they have a host that they've infected. Otherwise, they're just kind of inert molecules, really, or complex kind of assemblies of molecules. But I think agency begins with the minimal unit of that we can say is alive, and that is a cell. And it could be a single-celled organism like a bacterium, or it could be a single one of our own cells. But that's where agency begins. And so the question is, well, what enables a cell to have that complexity of response, because one of the interesting things about cells is they don't respond like simple automatic machines where, you prod them one way and you know exactly what they're going to do. You often do with a single cell. You certainly don't with an organism like us. But even single cells don't always respond in the way you think they're going to. Cells that look identical can actually do different things because, in fact, they have internal states of some sort that distinguish them. And the cell is making the decision of what to do based on reference to its own internal state, in a sense, just like we do, as well as the information it's getting from outside. So those are the kinds of questions that I think this notion of agency needs to address. And what I try to do in the book is to break it down to say, well, what are the ingredients that will make an entity an agent? Where do they stem from? Things like memory, things like a representation of their environment, a sort of simple model of their environment, which even single cells have, that enables them to kind of make predictions. Obviously cells don't think this, or at least I don't think cells think this, but in a sense, we can imagine them kind of thinking, well, if I act this way, maybe this will happen. If I act this way, maybe that will happen. This seems to be the best option. So that's putting it very anthropomorphically, but cells are really making choices, on that kind of basis of sort of imagining what a particular choice will lead to.
In the world of synthetic biology, how can we leverage some of the concepts you talk about in this book for things like therapies or other types of products that could be useful?
Right. Well, if we go first of all just to the idea of, medicine generally, then it seems to me that if we recognize there is this hierarchy of levels and no privilege level within biology, what we need to identify in order to figure out what we should do to cure a disease, say, is what is the real sort of causal level? What is causing the disease? Is it something that's coming basically from some mutant gene, or is it something that's a dysfunction of the immune system, or is it something else? What is the right level to intervene? If we do that, we have more chance of making a difference. But with synthetic biology, in some ways, so synthetic biology is this idea of that we can actually, in some sense, design organisms. And that comes with a very sort of traditional engineering perspective that seems to, give some credence to the idea that actually organisms can be thought of as, with circuit diagrams and as a kind of machine that we can, twiddle with here and there. What's interesting is that approach has worked in some ways. It's worked pretty well for engineering bacteria. We can design bacteria to do particular things that they don't do naturally, stick in a few genes perhaps, and it often seems to work quite well. It's proved much more difficult doing it with cells like ours because they don't work in the same way. They don't have this more deterministic way of working. They have these fuzzy rules. So it's much harder to make an intervention that has a predictable effect on what the cells will do. There are some people who are starting to make headway in applying synthetic biology ideas to so-called eukaryotic organisms like us and to more sort of multicellular structures, but it's proving much harder. And it's some people are thinking, rather than trying to do it from the bottom up, if we're wanting to use synthetic biology to make something multicellular, maybe a synthetic organ or something like that, maybe what we'll need to do is to do it more sort of through trial and error by, sort of tweaking the cells a bit and seeing if they build something different and then tweaking them a bit more to see, and perhaps using AI techniques to assist those predictions. But it'll be far harder to do it by design from the bottom up intrinsically because the rules are fuzzier. And so we might need to do it more through a process of trial and error.
Philip Ball, thank you so much for writing this book. I learned a ton, and thank you for coming on the podcast to talk about it.
Thank you, Andrew. It's been fun. Thank you.